Before we started discussing inhibitors, this is the last topic of Unit 1. The first type of inhibitor that we are going to discuss will be reversible inhibitors. Reversible inhibitors will bind in a non-covalent fashion. They can be removed from an enzyme. So that is the idea behind, that is the definition behind a reversible inhibitor. If it is reversible, it can be undone. You can reverse the process. A competitive reversible inhibitor is an inhibitor that competes with the substrate for the active site of the enzyme. And you can reverse the inhibition of the enzyme. A non-competitive inhibitor will reduce the efficiency of the enzyme without affecting enzyme-substrate binding. So, oftentimes this gets confused with allosteric inhibition. They are not necessarily the same thing. And this is actually an important distinction specifically for Cambridge. So, oftentimes they will ask you for specific definitions to define something specifically or to name the type of inhibition. And if they give you a type of inhibition that is an allosteric inhibition, you need to say allosteric inhibition, not non-competitive. Or if they give you a type of non-competitive inhibition, you must be able to say that it is non-competitive. A non-competitive inhibition is a type of inhibition that reduces the efficiency of an enzyme without affecting the enzyme substrate binding. So it does not affect the active site and substrate ability to bind to that active site. Allosteric effects are often equated with non-competitive inhibition. It is its own classification. Activators and inhibitors are both included as allosteric effectors. Allosteric effectors bind to a protein somewhere other than the active site and change the 3D conformation of the protein. It changes the overall 3D shape. And as we've discussed, the 3D shape is what determines the function of the protein. If a protein changes shape, it doesn't do its same job as when it had its original shape. The shape is what matters. The active site has a particular shape for a particular substrate. So, there is also a process called uncompetitive, as shown by this process here. Uncompetitive binds to the enzyme substrate binds to the enzyme substrate complex. But you're not really tested on that. Uncompetitive is simply that. It doesn't compete and it doesn't change the binding of enzyme substrate. Enzyme substrate binding has already happened. So it is not non-competitive and it is not allosteric, because the enzyme substrate complex has already formed. Allosteric and non-competitive both happen before the enzyme substrate complex has formed. Irreversible inhibition is just that. It cannot be undone. It is permanent. It will bind covalently to the protein with permanent effects. This is not the same thing as denaturing the protein. Denaturing the protein is changing its three-dimensional conformation in such a drastic way that it cannot change back to its original shape, either by making it too cold, too hot, too salty, not salty enough, too acidic, not acidic enough, too basic, not basic enough. Change the conditions too far in any direction, and you change the 3D shape too far so that it can't return to its original functional shape. You break it. You bend it too far and it snaps. Or you bend it too far and the shoelace comes untied. You pulled just a little too hard. That's denaturing. In irreversible inhibition, you bind another substance, an inhibitor, covalently to the protein, permanently altering its shape. You have not changed the shape of the protein by changing the environment. You have added something to it to change its shape and permanently bound that something. So this is competitive versus non-competitive. This is competitive. Here we have an enzyme, E, the substrate S, and the inhibitor I. The kinetic of the inhibition, the kinetics of the inhibition, or the constant of inhibition rather, Ki, is a reversible reaction as shown by the back and forward arrow. Shows and creates an enzyme inhibitor complex. Usually this is because the inhibitor has a similar shape to the substrate. A similar enough shape. Because the enzyme is the active site is designed to accept a shape, not a specific molecule, but a specific shape, which is recognized. A molecule is recognized by its shape. So something with a similar shape or a similar section of groups can be mistaken for it. So an inhibitor feels like the substrate and can therefore bind to the active site instead of the substrate. So here we have the enzyme inhibitor complex. Here is the inhibitor and here is the enzyme. This is this part of the reaction over here. Here is the enzyme inhibitor complex. Here we have the enzyme and substrate reacting in a reversible reaction, forming the enzyme substrate complex, which typically will move forward in an irreversible reaction to create the enzyme and products. This is the reversible enzyme substrate part of that reaction. The product part is not shown here. So this section is shown here. Substrate enzyme, ES. Enzyme substrate complex, ES. Reversible reaction. This is the reaction moving to completion over here. That's all that competitive inhibition is. This is a chemical equation. This is a graphical image. This is showing the imagery of it. So this is non-competitive inhibition. An enzyme, a substrate, and an inhibitor, the same starting points that we had before. You have the interaction between the enzyme and the inhibitor, and you end up with the enzyme inhibitor complex over here. But you can still have interaction between the enzyme inhibitor complex and the substrate to form the enzyme substrate inhibitor complex over here. On the same fashion, you can have the enzyme substrate react to form the enzyme substrate complex over here, which can react to form the enzyme and product just as it could before. But the inhibitor is still around, floating around. And then you have the interaction between the enzyme substrate complex and the inhibitor, forming the enzyme substrate inhibitor complex again. And this cannot react. This cannot move substrate to product. This cannot convert substrate to product. Enzyme cannot convert substrate to product when the inhibitor is involved. This is the graphical representation. Enzyme and substrate react in a reversible fashion to create the enzyme substrate complex. This can move to completion. Enzyme and inhibitor can form the enzyme inhibitor complex. And this enzyme inhibitor complex can react with substrate to form the enzyme inhibitor substrate complex. The same way this enzyme substrate complex, if it doesn't move enzyme substrate complex substrate to product, this inhibitor can react with ES to make ESI. ESI cannot change substrate to product. That is how a non-competitive inhibitor works. It prevents the movement of substrate becoming product. Substrate cannot become product. Even though it can still bind to the enzyme, it cannot become product. An inhibitor can still bind to the enzyme at any time. It does not affect the ability of substrate to bind. That is non-competitive. They are not competing. There is no competition here. They are not competing. This is simply preventing anything from happening. It doesn't work anymore. So now that we understand those reactions, we need to move forward and understand how they'll affect our equation. The equations that we went over last time, the Michaelis-Menten plots. Since our Km, our Michaelis-Menten constant, reflects our enzyme affinity, how readily a substrate will bind to the active site of an enzyme, how readily how readily the enzyme-substrate complex will form, how easily those two can fit together. An inhibitor that allows binding as normal but prevents the enzyme from actually functioning, which is a non-competitive inhibitor by definition, will only lower the maximum velocity of action, of enzyme activity. But the Michaelis-Menten constant will be maintained. The enzyme affinity is unchanged. Enzyme affinity is enzyme-substrate binding readiness. An inhibitor that does not change how readily enzyme binds to substrate will not change Km. Non-competitive inhibitors will not change Km. They will lower Vmax because you are not moving substrate and changing it into product. You are not converting substrate into product. You are making that reaction slower. You are slowing it down. You are lowering the maximum velocity. An inhibitor that prevents a substrate from binding to the active site, effectively lowering enzyme affinity, will raise Km, because remember, the Michaelis-Menten constant, Km, is inversely related to enzyme affinity. A high Km means a low enzyme affinity. A low Km means a high enzyme affinity. So an inhibitor that prevents enzyme-substrate binding lowers enzyme affinity, raising Km. Remember, Vmax is not tied to Km. If you keep adding enough substrate, Vmax can still be reached. Vmax is unchanged. This inhibitor that does this is a competitive inhibitor. A competitive inhibitor will raise Km and leave Vmax unchanged. Vmax will be unchanged and Km will be raised by a competitive inhibitor. This is also why we make the distinction between non-competitive and allosteric. Because allosteric affects whether or not substrate will bind to enzyme. Allosteric changes the shape of the active site. If the shape of the active site changes, the substrate cannot fit. If the substrate cannot fit, then the active site has a lower affinity. So an allosteric inhibitor will lower enzyme affinity, thereby raising Km. and lowering maximal velocity by inhibiting enzyme substrate complex formation and therefore the progress of the reaction. So allosteric inhibitors have a different effect on enzyme kinetic equations than non-competitive inhibitors. That's why Cambridge needs you to make the distinction. Not just definitionally, but graphically as well. So this is the graph. This is the normal Michaelis-Menten plot with no numbers. There are no numbers here, so you can't say high Km, low Km. You can't make that distinction without any real numbers. And you can't say high or low without two things to compare. Because the number 8,000 could be high or it could be low. If you compare it to 9,000, it's low. If it's over 9,000, then it's Goku. Yes, I know, I said I wouldn't make as many jokes during these review sessions. I lied. That's not the first time, it won't be the last. So, a normal enzyme Michaelis-Menten plot is this black line. A competitive inhibitor has the same Vmax as substrate concentration increases. You can reach Vmax. But Km, which is 1/2 Vmax, the substrate concentration at 1/2 Vmax, y equals 1/2 Vmax, x equals Km. The x value at 1/2 Vmax is higher than the Km of the black normal enzyme. This one, the 1/2 Vmax, is here. So here is the Km for the black line, here is the Km for the red line. The Km is higher. It raises Km. Higher Km, lower enzyme affinity. For a competitive inhibitor, same Vmax. The blue line is a non-competitive inhibitor. It prevents the reaction from going forward, but it does not change the affinity of substrate to enzyme active site. So Vmax is lower. Vmax is here in the black line and here in the blue line. 1/2 Vmax here is about here. That makes it that number right there. And there versus there. They have the same Km. Their Km value is the same. The blue and the black lines' Km values are the same. The Vmax is different, Km is the same. The enzyme affinity is the same. Their Vmax is different. That is a non-competitive inhibitor. If an allosteric inhibitor were in play here, both of these numbers would change and both of them would be different. It would be some combination of the two. It would be like in between these two things. That's what it'd end up looking like. It would be weirder looking. So you shouldn't be expected to estimate that look. But you should be able to explain what's going on. If they told you to describe and explain a graph, your explanation would be the allosteric inhibitor is affecting the shape of the active site of the enzyme, changing the affinity of the enzyme, therefore therefore changing the Km value. And it is preventing the reaction from proceeding, therefore lowering the maximal velocity of enzyme activity. So both Km and Vmax are affected in an allosteric inhibitor. Understood? So let's talk about allosteric effectors briefly as well. This is a graphical representation of how allosteric inhibition and activation works. This is highly simplified. Highly, highly simplified. The enzyme is this dude right here, the dark gray dude. An inhibitor will enter the allosteric site and change the shape of the active site of not disallowing a substrate from entering the active site. Without the inhibitor, this substrate would fit perfectly into the active site and the reaction may proceed. In an allosteric activation, the active site does not allow the substrate to enter. The activator enters, changes the shape of the active site, and allows the substrate to enter. This occurs because the allosteric site is the non-active site attachment point for some other binding protein, some other binding substance. It can be ATP, it can be a poison, it can be any number of other things. This inhibitor or activator attaches to the allosteric site, and through the interaction of its bonds with the bonds of the tertiary structure of the protein, the three-dimensional conformation, all the same things that make up the three-dimensional shape of the protein. Hydrogen bonds, hydrophilic and phobic interactions, ionic bonds, all of those things. Through a combination of those effects, shifts within that protein the overall shape. And when one part moves, another part moves, and another part moves, like a series of cogs and wheels, like a Rube Goldberg machine that then opens up the active site, or closes it down, or curves it, or opens it like an iris, or something. It's through the interaction of bonds throughout the entire protein macromolecule. It changes the interplay, the delicate interplay of bonds. It is always through the delicate interplay of bonds. And naming those bonds can get you points. So be sure that you go back and review those bonds again. Irreversible inhibition. Irreversible inhibition is a class of reactions that is similar to all of the reversible inhibitions, except for one more step. The dead-end complex formation. So here is a competitive or a non-competitive. This actually is a competitive, and this one is a non-competitive. Irreversible inhibition. So in a competitive irreversible inhibition, there is a back and forth. There is a reversible reaction here where the enzyme and inhibitor can bind to each other, and if it is gotten too fast enough, you can dislodge the inhibitor before a covalent bond forms, making it permanent. If you don't get there in time and remove the inhibitor, a covalent bond will form, and you get the enzyme inhibitor dead-end complex. Dead end. That's it. There is no way out of that blind end. You're done. That enzyme is worthless now. It is not worth the paper it's printed on. It is now wasted energy. This is the non-competitive one. The enzyme substrate complex has already formed. An inhibitor binds to this enzyme substrate complex. And that is still reversible until a covalent bond forms, forming the enzyme inhibitor dead-end complex. Or enzyme substrate inhibitor dead-end complex is also possible here. Now, if the inhibition does not take place, all of this can still happen. The enzyme substrate complex occurs, and the enzyme and the substrate is converted to product. Either broken down, built up, or inverted, or what have you. Now, there are many commercial uses for this, but I don't think those are tested. I don't recall which ones might be tested. Irreversible inhibitors, they do come in all the same flavors. Now, immobilization of enzymes. Immobilization of enzymes. Immobilizing something is making it unable to move. An immobilized enzyme is an enzyme that is physically attached to a solid support and a substrate is passed over it. You make it so that it cannot move. The enzyme cannot move, and the substrate is passed over that enzyme, and the reaction is allowed to proceed. Immobilized enzymes. This becomes important more in paper 3 than in paper 2. I have seen some paper 1 questions on this, however. Now, there are a number of advantages to attaching enzyme to a solid support. You can use a single batch of enzymes multiple times instead of making a solution with enzyme in it and then pouring in your substrate and then dilute and then distilling out your product. That is chemically costly, time-consuming, and you waste your enzyme product because usually to distill something requires boiling it, and that might denature your enzyme, so problems. But if you have enzymes attached to a card, for example, you can just dip in the card, wait 15 minutes, then pull out the card. Now, you can also stop the reaction rapidly by just removing the enzyme. So you pull out the card. Oh, I wasn't ready yet. Oops. Or you just fish out the bead. If you did an alginate bead and dropped it in, you just fish it out. And then you just pour off the unreacted enzyme, or the unreacted substrate. Immobilizing enzymes will also typically stabilize them, and you don't have to, as I said before, try and distill out your enzyme from your product. If you're attaching it to a solid object, you can store it for longer, and you can know how much you need to order. So commercially, it is very useful to us. These are some examples of the shelf life and the denaturation rates of enzymes that have been immobilized. When you try and denate an enzyme that has better activity and better recovery when it is bound, or immobilized rather. So these are just some examples. You should be able to read this simply by reading the axis. The y-axis is recovered activity percentage, and the x-axis is temperature in degrees Celsius. And these lines are coded by type, so immobilized tannase and free tannase. And the activity is shifted to the left here and shifted to the right here for the immobilized, shifted to the left for the free. And if you're asked, for example, to describe and explain the results of this experiment, you would describe that 3-tannase at this temperature has similar levels of activity. And enzyme 3-tannase peaks at 50 and immobilized peaks at 60 something. And you would use multiple data points. Always use as many data points as you can read. Use all the data points available to you when you are told to describe something. If you're told to explain something, why is this graph looking this way? Well, immobilized tannase has a larger range of has a wider range of active temperatures, which indicates that it is more resistant to denaturing at higher temperatures because it is bound to a whatever they told you in the question stem that it was bound to, alginate beads or whatever. The process of binding to alginate beads likely makes the tannase more resistant to being denatured, or makes the 3D conformation more stable. and makes the active site of tannase unchanged across larger ranges of temperature, which means that enzyme affinity is unchanged over a larger range of temperature. And that is why you see this spread. Free tannase, the reverse argument is that free tannase has a lower and it has a narrower range. And that is the explanation. That is what explain means. That is how you would explain those results. Describe is read the graph and basically put what this graph shows in words, data point by data point, and describe the trend. It begins here, rises to here, and then drops, and then falls here. And the difference between this one and this one, this one and this one is such and such. Explain is it happens because of this reason. The active site is such and such. That is describe and explain. And usually questions like that are worth 3 to 5 points. 3 points is 5% of the paper too. 5 points is what, 7%? Yeah, something like that. 7, 8%, 7.5%. No, 8%. I can't do math like that in my head right now. Leave me alone. All right. The same idea as pH here. It all has to do with the active site, enzyme affinity, and the three-dimensional conformation of the protein. That is the explanation for why it looks like this. Immobilizing it protects it from changing shape, making it more stable over a wider range. So the methods of immobilization. Films and coatings. Films and coatings. The enzymes are on a layer or a thin piece of material, and usually this is for like biosensors and stuff. This is when you need to do tests, when you need to detect something. So you're looking for the presence of something, and you put a sample onto this film, and by its presence, it will react, and you will see some sort of effect occur. And this way, there's usually multiple enzyme reactions going on there. Selective adsorbents. When you immobilize an enzyme in a matrix and then pass a substrate solution through it, you come out on the other end, your product solution. This is often useful for filtration. A controlled release. This is sort of when you're keeping an enzyme from functioning until it's ready. But this is really more of you're holding it and then dissolving that and it becomes free enzyme. It's bound enzyme and then it becomes free enzyme. So it's not controlled, but it's delayed. This is when you have time-release medications and the like. Now, these are some practical examples. This one comes up sometimes on the Cambridge Paper 3, and sometimes it's used as an example in Paper 1 or Paper 2 as a question stem. Milk that contains lactose is passed through alginate beads that have a bound lactase enzyme, which is used to break down the lactose and glucose, the disaccharide that is lactose. And usually they will ask you they will use this as a way of asking you about the immobilization graph. They will usually use this as a question stem to show you the graph of free versus immobilized enzyme, or use this to show you the graph of enzyme reaction over time, or show you rates of reaction between two different forms of lactase and ask you to describe and explain the difference between two different forms of lactase. And you can explain that by the enzyme affinity of the two different forms of lactase. If one is more enzyme affinity than the other, you can explain that through Km and Vmax. This is a urine dipstick test. Usually the one that we're interested in is the one for diabetes. And this is actually two chemical reactions, the glucose oxidase and the glucano delta. Yeah, I think it's delta lactone, gamma lactone. I don't remember which Greek letter that was. And hydrogen peroxide. So the glucose converts, the glucose oxidase converts glucose and oxygen into the D-glucura, blah, 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 and peroxide. And the other one is a peroxidase that converts H2O2 and that converts H2O2 into oxidized chromogen, which is colorful. And it changes the color based on the amount of H2O2 that it gets converted. So you may get a dipstick question that's used as a stem to ask you questions about, let's say, the different color changes versus concentration of glucose. So glucose concentration versus a colorimeter reading, an intensity of colorimeter reading. And you are to describe and explain that graph. So as colorimeter reading increases, glucose concentration increases. It's like a 1-to-1 ratio. What is happening here? Okay, this is this, and you are getting, you need to explain or describe that graph. So often these will be useful as question stems. So, these are the start of my slides from Unit 2. The microscope stuff. You can see with the naked eye things all the way down to about 100 micrometers, a tenth of a millimeter, 1/10 of a millimeter. That's the size of maybe the largest tardigrade. Light microscopes can also show you things that are that size. Is the width of a human hair that? I don't remember. All right. So a light microscope will bring you all the way down to around 100 nanometers for resolution. I mean, like, reliably. But your definite hard limit is 200 nanometers. At that point, the physics are against you. More on that on the slide that actually covered it. An electron microscope can cover all of this range of stuff. And an electron microscope can cover everything all the way down to fractions of a nanometer, but it cannot go down to the atomic level. There are microscopes that can do that, but they are not tested on Cambridge exam. Those are newfangled, fancy stuff. That is new science on the very, very edge of the frontier. So I want this equation burned into your psyche, and I have tested it so many times, and I really hope, really, really hope that it is still rattling around in there. The magnification equation. The magnification equation is magnification equals the size of the image divided by the actual size of the specimen. So, let's say that there is a beam that I blow up to the size of 1.5 meters, and that beam itself is 1 centimeter. And I want to know the magnification of my image. The actual size of the specimen is 1 centimeter in diameter, the beam. And the image size is 1.5 meters, measured along the same diameter. Now, I already have a problem with this because when I gave this example, I hadn't told you to stop using centimeters already. So, oops. Whoops. Sigh. All right. So already, I need to convert my units. I convert centimeters to meters. I multiply the meters by 100 centimeters, and I divide by 1 meter, because 100 centimeters is equal to 1 meter. This is the same as multiplying by 1. These are the same number, and 1 divided by 1 is 1. Multiplying by the same number does nothing. I am merely converting units. So 1.5 meters is my size of image, 1 centimeter is my actual size of specimen. I multiply by my unit conversion factor, 100 centimeters per 1 meter. Now, this leaves me with 150 meters times centimeters over 1 centimeters times meter. The centimeters cancel out, the meters cancel out. I am left with 150 unitless. By definition, by convention rather, not by definition, by convention, I use an x. X 150 times 150. That is my magnification in this example. So, my magnification equation is magnification equals size of image over actual size of specimen. Mag equals I over A. M equals I over A. M equals I over A. That will always be. It will always be. And you can do algebra to move those letters around. Total magnification is when you have multiple lenses, like in a light microscope, which has an objective lens that you look through, and a objective lens and an eyepiece lens. The eyepiece lens that you look through and the objective lens that you rotate around. So whatever the magnification of each one of those is, you multiply each one. So the first one times the next one. So if the magnification of the eyepiece is 10, and the magnification of the low power is 4, then the total magnification is 40, because 10 times 4 is 40. If the, for you guys, the red one on the microscope is the scanning power, the blue is the blue is the high power, the yellow is the low power. Yeah. So in this example, I had low, medium, and high, because that's just the thing I found, because I stole it. Borrowed for educational purposes. Now, the low power equivalent here is the scanning power on our microscopes. The medium power is the low power equivalent on ours, which is the yellow one. And the high power is the high power on ours, which is the blue one on our microscopes. The low power is the scanning, the red one on ours. So, red is 4 times the objective times the eyepiece, 10, 40. The low power, yellow, is the medium here, of 10 times the eyepiece, 10. 10 times 10 is 100. The high power is the blue one, which is 40 times the eyepiece 10, which is 400. So that is how you get total magnification. That is some videos that I had for you, and this is how we break down light and electron microscopes. These are the pieces of microscopes. And this is much more important for papers 1 and 3 than 2. Now, this is the diagram for the light microscope. This is pretty much somewhat what yours looks like. It is not exactly what yours looks like. So you have the ocular lens, the eyepiece, Yours rotates here to allow people to see if you need to share. You have a rotating revolving nose piece. You have 3 lenses. You also have space for a 4th oil immersion lens, which we do not use. We do not have those lenses in place on your microscopes because they're expensive and we don't want you to break them. And you don't need to use them anyway. And also we don't have the oil, so no, on all counts. And the scanning low and high power. You have a stage which supports your specimen. You have a mechanical stage clip. And you have actually a coaxial knob that moves that stage clip on an x-axis and a y-axis. And then you also have a coarse focus, which raises and lowers the stage a lot, and a fine focus, which raises and lowers the stage a very minuscule amount. If you are on anything other than scanning power, you never touch the coarse focus. The coarse focus is never ever touched unless you are on red scanning power. And I'm glad this is recorded for posterity. The coarse focus knob, the big knob, you never touch unless you are on the red scanning power. Because if you raise the stage and touch the lens to the stage, crack, break, very expensive, and it's coming out of your pocket. You break, you buy. Now, there is also an iris diaphragm, which regulates the light coming in. There is also the projection lens, which sends light through the iris diaphragm, up through this lens, into a series of mirrors in here, and then into your eyeball. The eyeball is the receptor here. You are seeing visions through your eyeballs. If you don't have eyeballs, you have bigger problems. Now, the arm is here. The base is here. When you move a microscope, you always hold it by the arm and the base. You never hold it one-handed, ever. Because if somebody scares you, you might go, ah, and drop it with one hand. With 2 hands, you're more likely to go, ah, but hold on with one, or ah, and hold on with the other. So these are precious. So hold with 2 hands. Very, very important that you do not drop them. You treat them special. You treat them very, very special. There is an on-off switch and a power plug. I hope you are familiar, since you are from the electronics age, with power plugs and on-off switch. Although, I would urge you to turn things off before you put them away, because it's rude to not, because the light bulb can still burn out, and replacing it is also, costly and time-consuming. So turn it off when you're done, and unplug it when you're done, because that's what you should do with electronic devices. You should turn them off and unplug them when you're done. So that is just good care of microscope. All right? An electron microscope, we do not own one of those, and we never will. An electron microscope, instead of using light to view an image, it uses electrons. Light microscope uses light to view an image. Electron microscope uses electrons to view an image. Unsurprising. The design varies, but they're all extremely expensive. So, this is a sample of probably a transmission electron microscope. Yeah. This is a transmission electron microscope example. The electron gun fires through a series of lenses. These lenses are not solid objects. Otherwise, the electron would just bounce off of them. It goes through a series of condenser lenses, which are merely electromagnets. which focuses the beam of electrons in a very specific directional pattern. And it fires this electron gun through the specimen. And then it fires it through a series of lenses beyond that, and then onto a detector, an electron detector. That electron detector then sends that series, that electron information as a series of ones and zeros to a computer that interprets it as a black and white image, as a series of yes or no. Did it go through something or did it not go through something? Did it bounce or did it not bounce? Yes or no. And that records the image. And so you have the final recording. Now, more on that after a break. Ready? All right. So here's the different types of microscopy again. Light versus electron microscopy. Light microscopes can give you images with their natural colors. This does not mean that all light microscope images will be colorful. This also does not mean that electron microscopes, which normally will naturally be black and white images, will only be black and white. There is such a thing as false color electron microscope images. You can add color to a black and white image. They do it all the time in order to accentuate certain features. They will even do it on the Cambridge exam when they have a point to make. A light microscope has a very large field of view, depending on how much you want to see and how large a magnification number you want to get. The larger your magnification, the smaller your field of view. Whereas with an electron microscope, you have a very limited field of view based upon the machine you are using. The machine itself has a specific limit on the size of the specimen you can put into it. And typically that means 100 micrometers is your maximum diameter of your field of view. Light microscopes are cheap in comparison to electron microscopes, which are very expensive, very expensive. And it takes very specialized training to prepare electron microscope slides. By the way, this has been a Cambridge question. State one advantage to using light microscope versus electron microscope, or state one advantage for electron microscope, or state why a light microscopic image was used for this specimen rather than an electron microscope. So that's why this is important. Light microscope, you cannot view living things in an electron microscope. They will be dead as soon as you try to view them, because you are firing beams of electrons at them if they are alive at that point. They are already dead most likely by that point, because the process of preparing them for the electron microscope has most likely killed them, because it requires that they be dehydrated completely. Completely dehydrated. You cannot view things that are full of water in an electron microscope. So, if you get a question that has a wet mount shown on it, explain one advantage of light microscopes over electron microscopes. An electron microscope needs to be dry, and you wouldn't be able to use a wet mount. You can't prepare a waterborne sample on an electron microscope. It will not have water in it anymore. It will be dead. If you want to see it move, you need it to have it. If you want to see it move, it needs to be under a light microscope. Now that said, you can kill it and put it under a light microscope. In fact, if it moves around a lot, it may be better if you murder it, because then it won't move around so damn much. And you can see it better. With light microscopes, your magnification maxes out around 600 times, unless you use an oil immersion lens, in which case you can get 1,000 times magnification. Your electron microscopes, you can get 500,000 times magnification. Your resolution. A light microscope has a hard limit on resolution. You cannot see things below 200 nanometers in diameter. But they will be very, very fuzzy regardless by that point. You won't see them at all at that size. But larger than that, they'll be quite fuzzy. Your resolution at best is going to be maybe 1/4 of a micrometer. Your resolution with an electron microscope is going to be much, much better. You will have a clear, sharp, crisp image at much larger magnifications. That is assuming that you're using a good computer image file. So if the image is blurry or your printer sucks, then no, you're not going to have a good image. It's not going to be very crisp. If your screen is bad, then no, you don't have good resolution. That's the screen's fault, not the electron microscopes. Or if your preparation was bad, you're screwed. So this is resolution. It's point-to-point discrimination. If you are asked to define resolution, it is the size of the interval between two separate points in an image. Resolving power is the capacity of an instrument to differentiate between 2 points. A smaller interval that can be distinguished means you have higher resolution. It's less grainy when you zoom in. We never care about individual dots. We always care about more than one. So this is the resolution difference between a light microscope and an electron microscope at the same magnification. These are the same magnification. This is the light microscope. This is the electron microscope. This is a clear, crisp image. You can see the bumps and the pits. You can see every dot and crevice. In the light microscope image, it is blurry, it is grainy. That is how you distinguish. They are both black and white. That is not how you distinguish. You can't tell if one is wet and one is dry. Huh? How would you? How would you tell? You can't touch it. It's a piece of paper. So, this is transmission versus scanning electron microscope. Transmission electron microscopes were developed first. Scanning electron microscopes were developed second. Scanning electron microscopes bounce electrons off the surface of an object and give you a scan of the surface. You see the surface of that object. A transmission electron microscope shoots the electrons through the object, and you see a cut. You see a section, a cross-section through the object. So these are plants. You now know what they are. These are animals. You now know what they are. So this is a light microscopic image. You can tell because there is color. There is graininess to the image a little bit. The resolution kind of sucks. It's two-dimensional looking. You see internal features of this cross-section. without too much internal detail of any individual cell, versus a transmission electron microscope. We see a cross-section of an individual bacterium, a unicellular organism. We see a lot of detail of the intracellular contents of this bacterium. These are mitochondria. We can see the cristae folds of the inner membrane. This is how deep you can go. This is 2 nanometers in size. This is the scale bar. This is a graphene layer, and this is a platinum. This is platinum protection, and this is a layer of graphene. There's actually 1, 2, 3, 4. 1, 2, 3. 3 layers of graphene on a silicon carbide substrate. That's how deep you can go with transmission electron microscopes. So you see a lot of internal structure, a lot of graininess. This is a scanning electron microscope. We see a lot of high resolution detail of the surface of a structure. And this is my favorite animal, the tardigrade, my spirit totem. Tardigrade in a light microscope version. Tardigrades, more tardigrades. Active, tune. Yeah, tardigrades. Tardigrades are awesome. Yeah. So, now, here's the deal. The major takeaway point there was barely brushed over. Magnification equals image size over actual size. Magnification equals image size over actual size. On maybe 80% of all the Cambridge exam paper 2s that I've seen, and well, maybe not every, 50% of the paper 3s that I've seen, there has been a question asking you to do magnification and calculate magnification and measure it. And that is why I have the shame on you rulers. Because you need a ruler for this exam. You need your own ruler for this exam. You need your ruler for this exam. Get a ruler for this exam. Get a good ruler for this exam. Not one of those ones that has like the hard to read numbers and notches. Don't rely on us to give you a good ruler. We will give you what we have, which you've seen what I have, and not the best. Get yourself a good, reliable ruler. The major takeaway point here, the thing that is tested the most often, and it's usually worth 2 points. Almost always 2 points. Sometimes it's worth 4. Is magnification. So here is the image. Figure 1.1 is an electron micrograph with transverse section of the palisade mesophyll tissue in the leaf of a flowering plant, Zinnia elegans. Name the features labeled A, B, and C. Okay, not as important for you right now, but I'm more interested in calculate the width of the vacuole across line XY. Show your working and give your answer to the nearest micrometer. Micrometer unit given there, mu m. Answer, ... micrometer there. 2. Worth 2. What does that mean? That is line XY. They are asking you to take a ruler and measure X to Y. And when you measure in millimeters. You always measure in millimeters, because every rubric I have ever seen always gives points for multiplying by the correct conversion factor of multiply by 1,000 millimeters per 1,000 micrometers per 1 millimeter. It's always image size in millimeters and the conversion factor is the conversion factor is times 1,000 micrometers per 1 millimeter. I hope this is in the shop.
Yes. Okay.
Now, let's take this one, for example. Here it gives us the magnification. It gives us magnification. And just to make my life easier, I'll just say that I measured that as like 7-something millimeters, or maybe just to make my life easier, not because that's what the actual measurement was on the exam. They know how much it is because they print the booklets. So they know exactly how much it is, and they always have a specific level of error that they are willing to accept within. And you must be able to measure using a ruler within the same level of confidence that they are willing to accept. So if you are more than 1 millimeter off from what they want, you are wrong. If your answer is wrong, they can sometimes give you partial credit as long as you did your math right. As long as your math is right, you can get partial credit. So it's important. The words here: calculate the width of the vacuole across the line XY, show your working, and give your answer to the nearest micrometer. When they say show your working and give your answer to the nearest micrometer, your answer matters. Both points can be given for the correct answer. If you don't show your working and you have the wrong answer, you will get nothing. If you have the wrong answer and you show your working and your working is correct, you can get partial credit. If your working is wrong and you have the right answer, you can actually get both points. When they say show your working and give your answer to the nearest micrometer, when they say something else, it changes. This specific phrasing means that your answer matters. Give your answer to the nearest micrometer. Nearest micrometer. I'll get back to that in a second. So, they give you the magnification times 7,000. So m equals 7,000. The image size is, I don't know, let's say I measured it as 7 millimeters to make my life easier. The actual size is what they're asking me. Calculate the width of the vacuole. They're asking me to calculate the actual size. So I don't know A. So I need to move things around here. I need to get A to the other side. Now I should have done this before entering my units. So A equals I over M. When you multiply A to this side and divide M from both sides, A equals I over M. That's just algebra. Yes?
I know a simpler way. Like you make a triangle and then you put I over M times A and you cover what you want to find.
If that helps you. This is how I do it. All right? You can share with your friends. Ask him. Him. And I leave that no names said because this is going in for posterity. All right? All right. So now, yeah. So now I'll convert. A equals 7 millimeters and 7,000. X 7,000. So now I need to give my answer to the nearest micrometer. The nearest micrometer. If I give my answers in millimeters, I will not get any points. And here, answer, dot, 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 micrometer. So if I write micrometer, it's wrong. Because micrometer, micrometer. Now if I had measured it as 69 instead, or as some other number, and I got a decimal place, because I used a calculator to punch it in, and I got a decimal place, and I put in that decimal, I could only receive partial credit because I didn't read the instructions. Nearest micrometer. Nearest micrometer. Nearest whole number. 1 micrometer. If I put in a decimal, I lose full credit. That is a free point for reading the instructions. That is how you answer this kind of question. That is the basic math. This is a similar question here. Show your working and give your answer to the nearest micrometer. Distance from X to Y. Same answer, same question, worth 2 points. Here, show your working and give it to the nearest micrometer. X to Y. Same question. Here, show your working. Express your answer to the nearest whole number. This time they're asking for magnification. And they're giving you, calculate the magnification of the electron micrograph in figure 1.1. Here, they give you a scale bar. A scale bar means that this line is equal to this number actual size. This is this many micrometers actual size. So whatever this image size is, I measure this image size. I measure this image size. And it is 10 micrometers actual size. A equals 10 micrometers. The image size is, let's say, 10 millimeters. That means the magnification is 1,000. Just making my math easy. It may not be 10. This is definitely not, because this is a smart board. It's bigger than that, for sure. All right? To the nearest whole number. If I put in a decimal or a fraction, I do not get full credit. I do not get 2 points. Show your working. Show your working and give it to the nearest whole number. The actual length of the cell between X and Y is 2 micrometers. They give you this actual size between these 2 points. And they're expecting you to measure the image size. And they want you to calculate the magnification. Now, I wish I could have found an example before we began again, but sometimes they will give you this particular phrasing. And sometimes they will give you this particular phrasing. And the phrasing is, you may lose marks if you do not show your working. That means, that is Cambridge code for, if you do not show your work, you will receive 0 points. All of the points come from showing your work. Every single one of the points. And when you show your work, the work that you are showing is image size Whatever you measured in millimeters, you write it down. You relist whatever they give you. Magnification, or actual size if they give it to you. You rewrite the equation. And then, once you've rewritten the equation, you fill in the blanks you know and multiply by the correct conversion factor. Almost always, it is converting millimeters to micrometers. 1,000 times 1,000 micrometers per 1 millimeter. And it is usually when they're asking you about image size over whatever. It's either M or A here. It doesn't, because it's always the same thing. Because whichever one you switch out, you're always searching for the same thing. Image size is always divided by one of these two to find the other. So, as long as you have the correct measurement, that's usually 1 point. Multiplying by the correct conversion factor with units conserved is usually 1 point. And, of course, conserving your units. Canceling out your units. And then writing your final answer correctly. And rounding it to the most appropriate number. So sometimes that'll be worth 2 points, sometimes that'll be worth 3 points, sometimes it's even worth 4 points. And when it is, it's always for correct measurement, rewriting the equation, multiplying by the correct conversion factor, and rounding correctly. If they asked you to put it in the round to the nearest, sometimes they may even say, round to the nearest 100 micrometers. And if they say round to the nearest 100 micrometers, the Cambridge rule of round up does not apply. You can round down then. If it's under 50, you round down. If it's over 50, you round up. You know, that rule goes back to normal. All right? Nearest 100. If they say round to the nearest 100 or round to the nearest 50 or round to the nearest 10, if they say round to the nearest and you don't, you have lost a point. A very, very easy point. When they say round to the nearest, that is almost always a point. If they say if you do not, you may lose marks if you do not show you're working, the actual answer you write down matters very little. It's always the work that gives you the most points. And you have to show your conversion, show your image size, label everything appropriately, rewrite the equation, and follow instructions. Read it and follow instructions. I cannot stress this enough, how important it is to read and follow the instructions. Most of the Cambridge exam can be answered and successfully completed with full marks by reading it and using the actual question to answer itself. There have been so many paper 1 questions where the answer was merely a paraphrase of the question itself. So many questions on paper 2 are a paraphrase of the question itself, with very little added knowledge from you. So many things on paper 3 are rote memorization. You just have to know the formula. You must read. You must read. And the image magnification questions are a perfect example of you must read. Show your answer to the nearest. If you don't do that, you lose out on perfectly easy points. I'm spending a lot of time saying this over and over again because I've seen it over and over again. All right. Now I can move on. Oh wait, no, I already showed that. My spirit totem. All right. This will probably all be edited out, this searching for the next thing. So this I went over before. But light microscopes have a hard limit based on the laws of physics. Details smaller than 200 nanometers cannot be resolved by light microscopes because visible light does not have a wavelength that is short enough to bounce off of things that small. So the shortest wavelengths of light are 500 or so nanometers, and 200 is less than half of that. So at that point, the visible light cannot reflect off of things that small. And so things that are that small are effectively invisible. Now, this is how you recognize animal and plant cells on microscope images. The difference between an animal and a plant cell is primarily in the differences between their organelles and their membrane, and their differences between their organelles and their membrane and wall structures. So an animal cell has observable features that are a centriole and centrosome, which is the surrounding area of protein structure surrounding it. Sometimes they may have cilia or flagella. And a plant cell will have a large central vacuole and chloroplasts. It will also have on the outside a cell wall, a rigid outer structure. Those are the observable features. When you are asked to state the observable features that distinguish this as a plant cell or animal cell, you should never, ever, ever state things that you do not see. Well, I don't see a cell wall, so it must be an animal. No. No. I see cilia, so it must be an animal. I see a centrosome and centriole complex, so it must be an animal. I see a large central vacuole, so it must be a plant. I see a chloroplast, so it must be a plant. I see a cell wall, so it must be a plant. If you are asked to indicate features you would expect to find if this were a plant, and they show you an animal cell, features you would expect to find if this were a plant, you would list things that you don't see, which are large central vacuole, chloroplast, and cell wall. If they ask for observable features, you can only list things that are in the picture. If you start listing things that are not in the picture, you will not gain points. You will not gain anything. You will fail. Observable means you can observe it. You can observe it in the paper in front of you. Not in your mind's eye, in your actual eye. So these are animal cells. You can tell that they are animal cells not by any observable feature. There is no observable feature here. that tells you these are animal cells. So here, what would you expect to find that would indicate that these are animal cells? If this were, since this is a light microscope, there's no way that you would see a centriole or a centromere, or a centriole or a centrosome, unless the cell was actively dividing. You can't see cilia here, and you can't see flagella here, because these aren't that kind of cell. Cilia and flagella are not present in every animal cell. So there's no observable feature here that indicates that this is an animal cell. But by a lack of plant characteristics, you can tell that this is a plant, this is an animal cell. It lacks a cell wall. So if you are asked, just blanket, if this is an animal or a plant, not by what observable features are there, then you can say with confidence this is an animal cell, because there is no cell wall here. There is no space between cells. An observable feature, I'll take it back. The observable feature is that there is no space between cells. The cells themselves are butting up right against each other with no middle lamella or individual spacing, intracellular spacing, which indicates a lack of cell wall. Here, we have observable features. We can see chloroplasts. We can see cell walls. So we know this is a plant cell. This is me being silly. And that was stuff. Yeah, that was me warning you not to be silly with the microscopes. And this was calibration. Good news is, calibration is not as hard as you think it is. Bad news is, calibration comes up both on paper 1 and very rarely on paper 3. They are very expensive. It very rarely comes up on paper 2. I don't think I've ever seen a calibration question on paper 2. It's a calibration. So, yay. Here's the deal. Calibration is merely finding out what a unit is worth, what an unknown unit is worth by comparing it to a known unit. I have no idea how much a goat costs, but I do know how much a hamburger costs. So if someone tells me that a goat costs the same as 500 hamburgers, I now know how much a goat costs. I have calibrated the goat. It's the same concept, except with rulers now. If I don't know how much a foot is, because, gosh, why would I? Imperial units are silly. Metric is so much better. I would, and I know how much a millimeter is, I would find out by taking a foot-long ruler and comparing it with a millimeter measuring device. And I would find that 1 foot is equivalent to 300 millimeters. I now know how much a foot is. I have calibrated my foot-long ruler. I have now calibrated that piece of equipment. The eyepiece gradical scale is my unknown. It is the goat, the foot-long ruler. It is the unknown. It comes in unknown, unmarked units. The stage micrometer is my known unit. It is my known unit. And by placing the stage micrometer onto my stage and measuring how many units of the stage micrometer units the eyepiece radical units equal to. And doing that fraction and finding that minimum fraction, I now have a conversion unit. I know that one eyepiece unit, one unit of the eyepiece radical, is worth however many units of my stage micrometer. And my stage micrometer is worth however many micrometers. That is how you calibrate an eyepiece radical. It is that simple. That's all it is. You are just comparing rulers to find out how much an unknown is worth based on a known. It is far less complicated than all the math seems to be. And when you think about it that way and reread this slide, hopefully it will come into context for you. Because the more I talk about it, I promise you, it will get more confusing if I continue speaking. I'm going to stop talking about conversion and calibration. Now, linear magnification is what I talked about before. It's the magnification equation again. So we're not going to go over it. We've already done it. Scale bars again. I've already gone over it with examples. When you see a scale bar, you measure the scale bar. If you measure anything other than the scale bar, you've wasted your time. The scale bar gives you something with a distinct beginning and end and a known value of actual size. A distinct beginning and end and a known actual size. Nothing else in this image matters except that scale bar. Nothing else. So, the image size is what you measure the scale bar to be. The actual size is what the scale bar tells you it is. And never assume the scale bar is lying about its weight. That's rude. The magnification is I over A, as it always is and always will be. Never eyeball it. Never start adding things going like this and going, neh, neh, neh. No. The definition of life. It must do the following things. It must eat. It must respire. It must excrete. It must transport. And it must reproduce. It must obtain fuel resources. It must use those fuel resources to make energy for itself. It must excrete waste products. And it must transport, actively move things from one place to another. And it must reproduce. It must be able to make more of itself. Now, because a unicellular organism is the smallest unit capable of doing all of these things, that is the smallest unit of life. That is how we fundamentally describe the structural unit of life. If it required 2 cells to do all these things, then 2 cells would be the fundamental unit of life. But because 1 cell is capable of all these things by itself, 1 cell is the fundamental unit of life. So long as a multicellular organism is comprised of similar units capable of all of these functions, it is a living thing. Even if, as a whole, it cannot do all of these things. So a mule, which cannot reproduce, is still a living thing because its cells can reproduce. Its cells can divide. Even though yeah, sure, that's all I'm going to go into. Why? So an organelle An organelle is not the smallest unit of life. An organelle is a unit within a cell. It is a membrane-bound structure within a cell. And an advantage of a membrane-bound organelle, which is sometimes actually directly asked by Cambridge, is that it can concentrate and isolate enzymes and reactants in smaller volume, thereby increasing the rate and efficiency of chemical reaction. Or it can contain and confine potentially harmful proteins and molecules in membrane-bound organelles, protecting the rest of the cell from their harmful effect. So, it can concentrate enzymes and harmful chemicals for its own use, or it can protect itself from those harmful chemicals by isolating them from the rest of the cell's contents. That is advantages of membrane-bound organelles. Remember this? Well, here it is again. Plasmodesmata is a newly boxed thing on this slide, because plasmodesmata is an intracytoplasmic connection between two cells. It allows the cytoplasm to communicate between two cells. Two plant cells next to each other, each one having their own cell wall, will share cytoplasm through plasmodesmata. Plasmodesmata is the plural. Plasmodesma is the singular. Do not write the plural when you are asked for the singular. I'm not sure whether or not you will be awarded the point. Remember the spelling of singular versus plural forms of these organelles, because spelling counts in Cambridge. My God, does spelling count? And spelling will always count. So, these are the organelles. In an animal cell, we will start with the things that are unique to the animal cell. The centriole is an assembly complex of microtubules. They occur in pairs. More information than that is nice to know, not required. They are surrounded by the centrosome. They are surrounded by the centrosome. That is not unique to animals. Actually, believe it or not, they are not unique to animals, the centrosome. Now, I apologize, I misremembered that again. The centrosome, previously, like two or so videos ago, I guess, I said that the centrosome was unique to animals. It is not. There are some very unique individual kinds of plants that have a centrosome, but there is no plant with a centriole. The centriole is unique to animals. These microtubule complexes are unique to animals. A centrosome, which is not shown here, is this haze stuff. It is a proteinaceous area around the centriole that contains and becomes the microtubule unit. It pieces them together. It's what builds from. These act as a nidus for the creation of microtubules during cellular division. This is where the spindle fibers form in cellular division. That is what they do. This is an organizing center. The centrosome is also the organizing center. But the centriole is a nidus for organizing. The microtubules themselves form from here. Now, cilium and flagellum are not shown here, but cilium and flagella I will actually discuss later. I have a slide later. I'll go over it then. That's great. So now to the stuff that is not unique to animals. The stuff that is shared between animals and plants. The cytoskeleton plays a supporting role in maintaining the shape of the cell itself, and also in the motion of the cell, and also in the movement of vesicles from point A to point B. Yes, animal cells have vesicles. They have many vesicles. They are just not a large central vesicle. Microtubules, intermediate filaments, and actin filaments are all types of filaments present in the cytoskeleton. Cytoplasm is the fluid matrix that exists as the liquid medium wherein all of the chemical reactions of the cell are, all the chemical reactions occurring outside the organelles are occurring. The cytosol is another name for the fluid part that is not the nucleus. Just the fluid part that is outside, not the actual organelles. The mitochondria. Mitochondria are not, I repeat, are not the powerhouse of the cell. If you write they are the powerhouse of the cell on the exam, you will have points deducted. It's written into the rubric on multiple occasions that if you say they produce energy, you lose points. If you say they are the powerhouse, you lose points. It's written in. You lose points for saying stupid things like that. It is dumb. It is wrong. The mitochondria are double membrane bound organelles with an outer membrane and a folded inner membrane. The folded inner membrane are folded with many intricate inner folds that are called cristae, that serves to increase internal surface area, to increase the number of cell surface membrane, of inner surface membrane reactions. Their primary function is cellular respiration, aerobic respiration, the conversion of fuel into energy for the production of ATP. ATP is not energy. If you say the word energy without mentioning ATP, you lose a point. If you say ATP without mentioning energy, that's fine, you keep the point. If you say ATP energy, you lose the point. So for the production of ATP, adenosine triphosphate. So their primary purpose is to produce ATP by aerobic respiration, to convert fuel, usually glucose, sometimes long-chain fatty acids, into chemical energy that is then stored in the bonds, high-energy bonds of ATP. ATP is an energy storage molecule. You must absolutely clarify. If you use both the term energy and ATP in the same answer, you must clarify that ATP is an energy storage molecule. You must distinguish those terms or you lose points. In fact, sometimes, if you are referring to respiration as the main goal and ATP production and that mentioning ATP as an energy storage molecule, sometimes that's actually 3 separate points. So it may actually be beneficial to you to make that distinction in writing. Make that distinction. Clarify. Clarify. Make that clarified. Make it clear. Now, It is a double membrane bound organelle. Sometimes that's worth a point. It has intermembrane folds called cristae. That's actually 2 points. Intermembrane folds called cristae, wherein aerobic respiration and oxidative electron transport chain occur. The electron transport chain occurs. But the electron transport chain is not part of AS levels, so I don't know whether or not you'll receive any points for that. It's just part of my explanation. It's really the only way I know how to put it in words for you. Right then. Now, on to, let's go to the nucleus. The nucleus is not the command center of the cell. I know it says it here. That's because this image is taken from the internet, because I can't draw. The nucleus is not the command center of the cell. The nucleus is the largest organelle and contains the genetic information of the cell. That is what you say. That is how you phrase it. It is the largest organelle, and it contains the genetic information of the cell. In chromatin, held within chromatin, which is comprised of DNA and histone proteins, bound into a 30 nanometer wide fiber, coiled fiber, is usually additional points. More on that when we discuss DNA again in Unit 3, in the Unit 3 review. The nucleolus, of which there can be more than one or none at all, is a specific site within the nucleus where ribosomes are made. It is a darkened spot. It is the site of ribosome production. Here, there is a high density of rRNA and proteins being bound together into large and small ribosomal subunits to be being ready to be exported out of the cell, out of the nucleus, being ready to be transported out of the nucleus. This is the site of ribosome production. It is producing rRNA and possibly binding it to proteins. The nuclear pore is the communication between the nucleus and the cytosol. Anything that needs to come in or out of the nucleus must go through the nuclear pore. It is a large structure. It goes through the nuclear envelope. The nuclear envelope is a double membrane. The inner membrane faces the chromatin, and the outer membrane faces the cytosol. The nuclear pore is continuous through both layers. The intermembrane space is not really helpful to you for any purpose in the Cambridge exam. Now, the ribosomes are composed of rRNA and protein. That is always 1 point. Anytime you're asked to describe ribosomes, they are composed of rRNA and protein. They are comprised of a large and a small ribosomal subunit, and in eukaryotes, they are 80S in size, and in prokaryotes, they are 70S in size. That may or may not be 1 point. It may actually be 2 points sometimes if they're asking you to compare prokaryotes and eukaryotes. So that's 3 points already just describing ribosomes. Now that was just their structure. Now I describe their function. They function in the production of polypeptides by binding to mRNA and allowing tRNA to bind codon by codon to the mRNA, anticodon to codon, in a complementary fashion. Right. So the endoplasmic reticulum serves a synthesis role. The rough endoplasmic reticulum is rough because it is studded with ribosomes. Describing the composition of ribosome gets you points. Describing that rough endoplasmic reticulum is rough because it's covered in ribosomes probably gets you points. Describing the role of rough endoplasmic reticulums, that it probably is for the synthesis of proteins, gets you points. Describing that smooth endoplasmic reticulum is a series of intracellular internal membranes that is involved in the synthesis of steroid and lipid compounds, gets you points. And it's involved in the manufacture of carbohydrates and lipids and steroid compounds. Those get you points. The next step in the process is the Golgi apparatus. The Golgi apparatus is a series of flattened membranes. Its job is to collect, package, and distribute molecules that are manufactured by the cell. It releases vesicles. It receives vesicles from the endoplasmic reticulum and then passes those vesicles from one membrane layer to the next membrane layer to the next membrane layer to the next membrane layer until it releases the last membrane layer as a Golgi vesicle, which may either be a transport vesicle to another part of the cell and transported along the cytoskeleton by transport proteins, or it may be sent out as a secretory vesicle. A secretory vesicle fuses with the cellular membrane, releases the contents of the vesicle into the extracellular space by a process of exocytosis. More on that when we discuss membranes very shortly. So, the lipid bilayer that comprises the cellular membrane, describing that is also worth points. Describing that as a lipid bilayer with a hydrophilic head on the intracellular facing side, a double layer of hydrophobic tails, and a hydrophilic head facing the extracellular end is worth points. Having membrane proteins of varying types and describing those types and being able to recognize the different types is worth points and has even been its own questions. You have been tested on them before. And in later review sessions, there will be practice questions that I will give you. Now, cisternae is a definition that is the blind end of an endoplasmic reticulum membrane area, internal membrane tube. Usually, when you get to a cisternae, this can pinch off to form an endoplasmic reticulum vesicle. Now, vesicles themselves. There are multiple kinds of vesicles. There are Golgi vesicles, there are transport vesicles. There are also two types of vesicles that are worth mentioning specifically: lysosomes and peroxisomes. Lysosomes are vesicles used primarily for digestion. They break down macromolecules and digest them and they also digest worn-out cell components. They're also useful in the destruction of cellular invaders. They're also very useful for the immune system to destroy engulfed pathogens. As part of the process of phagocytosis, a phagocytized particle will be engulfed and be held within an endocytic vesicle, and that vesicle will fuse with a lysosome, forming a phagolysosome. This is something we'll cover in more detail in Unit 5. This phagolysosome is part of the digestive process. That digestive process will continue until the thing is dead. And that mixed up bag of dead parts will then be either absorbed, released, digested, used for energy, sent to the mitochondria, or just excreted as waste. The peroxisome is a vesicle that contains enzymes, especially peroxidase enzymes, that carries out particular reactions to detoxify the cell, also removing specifically hydrogen peroxide, turning hydrogen peroxide into H2O and oxygen, so that you are taking reactive oxygen species that could damage DNA and keeping them from becoming those reactive oxygen species, just making oxygen and water. Oxygen your body, your mitochondria can use, water that is part of your cellular makeup anyway, as opposed to H2O2, which is just plain old dangerous. So that's part of the peroxisome's job. It detoxifies harmful molecules. It is also useful in breaking down long-chain fatty acids into medium-chain fatty acids. This is probably not useful for the Cambridge exam, but it might get you another point if they include it in the rubric. Maybe. They may not include it in the rubric. It might not be included. And you may not ever see a question about peroxisomes. So, just useful information for you in the future. This is the plant cell. A plant cell has all the same stuff as an animal cell, except it does not have centrioles. Like I said, some plant cells can have centrosomes, which is a microtubule organizing center. But it does not have centrioles. They have a nucleus, they have a nuclear envelope, they have a nucleolus, they have chromatin, they have a nuclear pore. They have all the same stuff. They have mitochondria, they have Golgi, they have ribosomes, they have endoplasmic reticulum, smooth and rough. And you must be able to recognize smooth and rough by its shape in a cartoon version. That is worth points. You must be able to draw the cartoon version of those shapes. That is worth points. Now, here is the unique stuff for plants. The large central vacuole, a large fluid-filled sac. It contains tonoplast, which is used to maintain turgor pressure. It also stores metabolites. It's also sometimes used to store in very certain types of plants things that are, well, poison to whatever might eat the plant. Things that would scratch up the animal's throat, like graphite crystals and things. Not that important. Not on the Cambridge exam. Excuse me. Right then. The cell wall, that's easy. The cell wall is composed of cellulose. If you are asked a question about the cell wall, sometimes they can mix Unit 1 and Unit 2. The composition of the cell wall is cellulose, beta-glucose, in long chains that is stringed together to form microfibrils, that form fibrils, that form fibers. And those fibers can be in layered patterns, or they can be in cross-hatched patterns. But it is a porous pattern. The cell wall is freely permeable to water. Freely permeable to water. It forms an outer protective surface that is rigid in structure and supports and protects the cell, You do not find cell walls in animal cells. When you are drawing cell walls on the paper 3, you draw cell walls as a double line. Always as a double line. When you draw two cells next to each other, you draw each of them with their own cell wall. Each of them has their own cell wall with a line between them. This line is the middle lamella, the middle layer. That is extraordinarily important. If there is no double line, there is no point. If there is no middle lamella, there is no point. That is extraordinarily important for all plan diagrams of cells. Anytime you are asked to draw a picture, or draw a plan diagram rather, of plant cells, they will always have a cell wall drawn as a double line. No shading. Do not start shading in these cell walls. That is wrong. Plan diagrams are not shaded. They have no shading. They are only lines. They are merely line diagrams. They only show a floor plan. And you make those lines as clean as humanly possible. Not like me with a whiteboard. A dry erase marker is not as good as a sharpened number 2 pencil. And when I say a sharpened number 2 pencil, I mean it. I mean you sharpen your pencil. Because you can also lose points for having a line that is thicker than 1 millimeter. You can also lose a point for having lines that overlap like this. You can also lose points for that kind of thing. You can also lose points for wiggly lines like that. So nice, smooth lines. But you draw what you see if you're drawing from a slide, a prepared slide. All right? That was an aside for paper 3 especially. Always, 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 cell walls are drawn as a double line. Always. Now, let's talk about chloroplasts. Chloroplasts, like mitochondria and the nucleus, are double membrane bound organelles. Within them, they also have stacks called grana. These stacks are stacks of thylakoids, which are chlorophyll-containing units. This is where the photosynthesis happens. You do not need to understand photosynthesis more than a very rudimentary level for AS level, because photosynthesis is A level. Respiration in mitochondria is A level. You only need to understand the very basics. For mitochondria, food in, ATP out. For chloroplasts, light and carbon dioxide in, sugar out. Sugar and ATP out. There's a little more to it, but I don't have the slide in front of me to read it off properly. So, this is my recap. This will be edited out. This is my recap chart of the different organelles and whether or not they are present in animals and plants. This is a transmission electron microscope of an animal. This is an animal because there is no cell wall. This is the plasma membrane right here. We can tell this is the plasma membrane because we see empty space. In a transmission electron microscope, there is no such thing as empty space unless there really is nothing there. Unless there really is nothing there, there is no such thing as empty space. Even the space within cells is full of stuff. There's always stuff there. The nucleus is here. This white stuff is euchromatin. This black stuff is heterochromatin. These are mitochondria. These grill marks are rough endoplasmic reticulum. The black dots are free ribosomes. This is a close-up of each individual cellular membrane. This is the lipid bilayer. And this is the other lipid bilayer. And this is the intercellular space between them. The black line is the hydrophilic head. The white